Okay, so we can start from where we stopped. So this is yesterday. Viruses infect bacteria, multiple phages, sometimes not invade, sometimes invade. And then we studied what happens when we had evolution of the bar size. And what we discovered is that if you have if you put together two different mutants and you put together a mutant, the one that survives is the one, or a mutant is able to invade if it has a birth size larger than the other one. Okay? So everything as expected. Okay, this is just looking at the sign where we really see this effect. Good. So then we talked about trade-offs, and we discussed how there is a large variation of the birth size, and how the birth size is connected to the latency period. And I think that the point that we arrived to was to say, okay, in reality, this birth size is some function of the latency rate. Okay, right? And good. And then the question that we were having is, okay, so in this case, what happens to the succession of invasions, right? This is the point where we arrived. And the last point we discussed is, okay, so suppose that we have a mutant and this mutant has some beta prime, and we have here beta prime minus e prime minus omega prime. So what happens to such a mutant, right? Where now we have beta prime and beta prime, and they are connected in this way. Okay? And so yesterday we learned something. So what should we do now to get this answer? If we want to know whether this mutant will be successful or not, what should we do? Yes, okay, we can have beta prime, but then first of all here we have n, and n is a dynamical variable. But then what is the assumption we make about n?
That the new stage enters when there is like a equilibrium state.
Exactly. So that the invader is rare. So we want to see whether a rare invader is growing. And the idea is that since the invader is rare, in this initial growth, which determines the future success or decline, there is no effect of the mutant on the population. So the trick here is that this equation is a mess because it's non-linear, right? Because there is n times v, and then is a solution that depends on the other population. But then we can simplify and say this is actually equal to star, which is a property of the, which is the population set by the resident phage. Okay, so what we have is that phi n star is equal to what was it? To omega. Sorry, the number anymore. I have to copy it. So phi n star is equal to omega beta plus omega over beta minus 1 beta minus omega. Okay. What is important here is that this population here is the population that is determined by the property of the resident phage population. Right? So this depends on beta and eta, which are the traits of the resident. Okay? So then we have this equation, which is linear. And so what do we do? Yeah, we have the matrix, and so we look at the largest eigenvalue. And the largest eigenvalue is what determines whether our population will grow or not. Okay? So when we look at, if we look at one of them, so when we look at this plot here, okay, this, you see that this concentration is the log concentration, it's linear in time. So this thing, right, this rate here, okay, I mean, this population is growing exponentially, right? Because the log of the population is linear in time. So this thing is some, let's say, lambda of the invader times time. Okay? And this lambda of the invader, okay, which is the rate at which the invader is growing, or if we go back, the rate at which the invader is declining, is exactly the largest eigenvalue of this matrix. Okay? So if we want to know whether the invader will grow, so let's call these things S. Okay? If we want to know whether this invader grows, right, what is the growth rate of the invader when it arrives in a resident population, this will be done by the largest eigenvalue. Okay? So what is natural to introduce here is a quantity that is exactly this right here, which I call S eta, S, let's say beta prime given beta, okay? Which is the growth rate of an invader with beta prime when the resident population is beta, okay? Is it clear what this object is? Is this quantity here? So mathematically is the largest eigenvalue of this matrix, okay? Of the matrix that is represented here. Biologically or physically is the slope of the growth rate of a rare invader with rates beta prime when the resident population is beta. Okay? So since we know, I mean, how to calculate eigenvalues, we can calculate this quantity. Okay? And this is exactly the eigenvalues of this. And except for a proportionality constant, which I don't care, this quantity is beta prime over beta prime plus omega minus beta eta. Okay? Now let's look at this expression again. So this quantity is the growth rate of an invasion. Right? Now let's suppose that we forget about this relationship and we look at the case where only beta changes, eta is constant. If that is the case, this term here and this term here are the same, and we get that this quantity is beta prime minus beta times something. Okay? So this is what we discovered earlier, right? This is the calculation we did earlier. That the growth rate of an invader, where the only difference is the bar size, is proportional to the difference of the bar size. Okay? Now, if the latency times are different, the situation is more complicated. Okay? So what we can do is to find the things for so the idea is that this quantity is telling us whether this population will invade or not. Okay? Good. So let's look how this quantity looks like. Okay. And what we can plot is the same plot as before. Right? So you think about what we did here. Basically, we were looking at the case where here in this plot, we were looking at exactly this quantity. Right? And in this case, this quantity was beta prime minus beta prime sum. Okay? And so you see that here you have beta prime equal to beta. And then above this value, beta prime minus beta is positive. Below this value, beta prime minus beta is positive, is negative. Okay? So now we can look at this more complicated case under the assumption that beta eta is beta naught, beta plus. Okay? And if we do that, what we find, blah, 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 okay, it's something more complicated. Okay? So this is exactly the plot of this quantity of S of beta time. And let's look at, let's try to understand what are the features of this plot. So first of all, this quantity is equal to 0 on this line. Okay? This is a very general feature, right? So whatever model we have, if we have some traits, theta prime and theta, okay, if all the traits of the invader are equal to the traits of the resident, there is no difference. So we have that S of theta is equal to 0. Okay? And this is exactly what we see on this line. But then in this case, there is something else. We see there is another line, which is somewhere here, okay, where this quantity is also equal to 0. Okay? Good. Now, let's do the same game that we were doing earlier. Let's see if I have the signs. Yes. Okay, so we can simplify and forget about the value and just look at the sign of these things, whether this is positive or negative. Right? So this is how it looks like. Okay? Okay? So let's look at invasions. Suppose that I start here. Okay? And suppose that I have a mutation here. Does it invade or not? No. Then I have a mutation here. Does it invade or not? Yes, exactly. So what do you think is going to happen over time? So if we wait a long time and we have this system evolve, what is going to happen? What happened here?
We reach the center.
Yes. We reach the center, and then in the center here, we're stuck. What does it mean we are stuck? It means that there is no mutation, okay? In this case, independently of how big it is. But there is no small mutation, if you want, that can invade this system. Okay? So this state here, how should we call this state? Okay? First of all, it's a state. Then it's something that is sort of stable, right? You expect that there are mutations and they don't affect this. And it's stable under what? It's stable under evolution. So these kind of states are called evolutionary stable states. Okay? The idea is that there are states, traits, values, that cannot be invaded by any mutant. Okay? To be more precise, this is a local thing. So we are assuming that we are here and there is no small mutation that can invade it. Right? In this case, it's global. There is, I mean, whatever mutation you don't invade. Okay? So what we see in this case is that we have something different, that over evolutionary time, what we expect is that this quantity here is sort of the outcome of evolutionary dynamics, in this case, independently of where we start from. Right? Is this clear? So now you can imagine of. Okay. So I'll show this. Okay. So good. So this is an important concept. Now, suppose that you want to calculate this value here, the value of beta that is cannot be invaded, the value of beta that is the evolutionary stable state. How do we find? Yes. One way is the intersection of the two lines. Or think about these things, right? This is telling me, I mean, these things has a very special form. Okay? We will see that, but we will see that this is more general. It's a very special form because this quantity here, it's the difference of something that depends only on beta prime minus something that depends only on beta. Okay? So when do you think that you cannot be invaded? What values will not be invaded? Right? It will be the combination of beta prime and eta prime, such that this quantity is the maximum. Right? So the property to have this feature here is if this thing is maximized. Right? Because if this thing is maximized, right, there is no other combination of beta and eta that can invade. Okay? So how do we maximize this thing? Well, we can, for instance, express this quantity with respect to, so we want to maximize this quantity here. Okay? So in this case, we can say, okay, we have beta of eta prime over eta prime plus omega. Okay? And what is this function? This function is, well, beta naught eta prime over beta prime plus beta naught omega plus beta prime. Okay? Right? Then to maximize this quantity, what do I have to do? Take the derivative, right? So I take the derivative of this quantity with respect to eta, and then what do? You set it to 0. Okay? Now, you do this derivative, and what you find is blah, 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 blah. What you find is that this is equal to 0. I can skip doing the derivative. Who wants me to do the derivative? Okay, you find it, and when you set it to 0, you find that the value of eta, such that this is 0, is equal to the square root of eta naught omega. Okay? So what you get here is that you get that this eta ESS is the square root of eta naught omega, and therefore beta ESS is beta naught over square root of eta naught omega plus eta. Okay? Now, let's comment a little bit on this. So, first of all, one property of this system is, as I say, is that the invasion, the growth rate of the invader is the difference of two quantities. Okay? It's the difference of something which depends on the invader minus something that depends on the resident population. Okay? Now, if you think about what we did when we talked about evolution, I was talking about a quantity called S, selection coefficient, and say that this was the fitness of one population minus the fitness of the other. Okay? So the idea here is that in this case, and this is a special case, we will see later, in this case, I can interpret this quantity as a sort of fitness of the invader, and this quantity as the fitness of the resident population. Right? Okay? And what do we expect over evolution? What we expect is that we expect the fitness to grow, right? We expect that over evolution, fitness will grow. And so, in particular, fitness will go to a maximum. And in this case, this quantity has a maximum, okay? Which is the one we calculated. Okay? So the idea is that in this case, you can interpret this as a fitness. And so somehow, this is an optimal condition. Good, first comment. Second comment is what determines fitness? If you look at this evolutionary stable state, what is determining the value of the evolutionary stable states? What are the parameters appearing here? Well, eta naught or beta naught, which set the relation between the birth size and the latency period, which is a sort of physiological constraint. So there is one important property, which is constraints. Right? So you cannot make a virus with a birth size that is too big if you don't make a latency time more period, in the same way as you cannot make an animal with wheels. Right? You have constraints. Okay? You have to operate with physiological constraints. This is one thing. Then there is another parameter that enters in this expression, which is omega. And what is omega? Yes, and who decides omega? Who decides omega? If you remember, omega is the rate at which we remove stuff from the experiment. So who decides omega? Us, right? Because we can choose to do another experiment where we change omega. So if you want omega, in this case, is the environment, right? Which is telling you another very important lesson, that fitness is not a property of an organism alone. Right? It's the property of an organism in a given environment. If we change omega, what we expect to change is to change this ratio here. In particular, if you want to give an interpretation of this, what happens if you change omega, right? What happens to the birth size? It will, the evolutionary stable birth size. If we increase omega, it will decrease, and the rate will increase. Why? Because the risk of omega, okay, why omega is setting this? Because if omega is large, there is a large probability that when a virus infects an host, these infected cells disappears. Right? And so the price to pay is that if you have a too long latency period, okay, then there is a high chance that when you infect a cell, you actually lose that infected cell. Okay? So if you have a large dilution, if you want, if the mortality of infected cells is large, it's better to be very fast. It's more efficient to infect and lyse fast rather than produce a large birth size. Okay? But the important message here is what evolution, the evolutionary dynamics, what we see as a change in evolution is a combined property of the constraints, beta naught and beta naught, and the environment, in this case omega. Okay? Questions about this?
Can you think of this rate as stress in
As
the
a?
environment, as a stress?
This omega, yes, you can think as a stress. Yes, I mean, it's a stress in some sense. So basically, I mean, if you want to give it to this an economic interpretation, omega is something that is related to your time horizon. Right? So basically, a large omega, so a large rate, means that your time horizon is short, because there is an high chance. And so then if your time horizon is short, what you should do is you should invest in infecting and lies fast, producing less return, right? So if your time horizon is short, it's better an egg today. If your time horizon is long, it's better a chicken tomorrow. Right? And these things is weighting your time horizon, right? So large eta means tomorrow, and large beta means chicken, right? And large latency period means tomorrow, this is small eta, and chicken tomorrow, while a small time horizon implies a small latency time, so today, and small burst, so egg. Okay? Good. Okay. Now, what is important also as another comment is that now we can interpret this as an optimality condition, right? We can say, okay, over evolution, I mean, the way we derived this was as an optimality condition, right? But, I mean, the interesting fact is that we didn't decide a priori what was this term that we were optimizing, right? So, the question we asked ourselves was not what evolution is optimizing, right? And try to build a quantity that evolution optimized. What we did was to set the rule of the game, so how individuals reproduce, and then we derive this quantity here. So this is an output of our things, it's not an input. Right? This is very different from economics, for instance, right? Where you think that you have a utility function or there is something you are optimizing. In this case, evolution does what evolution wants. Okay? Is this clear? Okay. So this is. Now I want to make this a little bit more general. But before this, I want to. I mean, with the code that I provided, you can sort of. I mean, you can derive all these things. But what you can do is that you can simulate evolutionary trajectories. So this is the kind of things. These are pretty short because I didn't run it for a long time. But so this is like you have initial phage. These are here. This line here. You have the initial phage population with some beta, and then what the code does is that with some rate you introduce mutants. Okay, and these mutants are a copy of the parent with a beta around a value of beta around the one of the parent. Okay, so you take the beta of a mutant from with a mean equal to the parent and the Gaussian distribution with some values. Okay, and what you see is that there are mutants that appear. So many mutants don't make it actually go to extinction, but then you see that there are some. I mean, for instance, in this case, there were two, right? Which by chance they had the same color, and these two probably have a similar beta, which is larger than this one. This is beta equal to 5, and in this case it's growing. So they grow, right? You see also the orange one is growing, but then this one probably is the one with the largest beta of all. So all the others are better off than this one, but then are worse than this one, so they go down. And so you see that then this stays around for a while, but then there is probably a mutant, which is actually here, right? Which is growing, taking over, okay? And then new mutant appears, etc. So another way to visualize this, is that you can look at the values of beta, you start from 5, and then you look at how long do the mutants stay. So for instance, here you have these small dots because there are mutants with a smaller value of beta that appear and then go to extinction. Right? And what you see is that you have these things that grow, and then you have a lot of mutations because they want to take over, And so you see this evolution towards higher values of beta. And if you look at the average value of beta in the population, you see this sort of evolution. Right? And you see these steps in this case because you have these sort of slow dynamics of taking over. So you stay there for a while, and it takes a while for ecological fields. Okay? So the question that I want to ask is now, if you see this system is a mess, okay? If you see this system and you look it from afar enough, it looks like something that is growing, okay? So the question is, suppose that we want to write some equation. What equation would we like to write? Suppose that we want to describe some evolutionary dynamics. What we would like to write is, so this quantity here is the average value of beta in a population, okay? So you take the abundance, relative abundance of virus I over all the viruses, and you look at the sum over J, beta J, xj. So it's the average value of beta in the population. What we would like to write is what is the variability of this beta over time. Okay? So if we don't care about the sort of evolution, I mean, we care, but we don't want to capture all this mess. What we want to capture is how does the trait, the important trait, in this case the birth size, change over time. Okay? The question is, how do we capture this quantity here? Now, I will not give you the full-blown derivation of that, but we can sort of try to think about this starting from this thing here. Okay? So suppose that we have a mutation, right? We are here. And we have a small mutation. Right? And then what happens is that this new mutation does something like this, right? The process we described before. There is a mutation, and this mutation sort of appears in the population. Okay? So how much do you think is going to, and now we forget about the variability within the population. Imagine that there is just one type here and one type there. Okay? So how it's going to change this delta beta? Right? What is going to be the change of this delta beta in some unit of time? Okay? To what will be proportional? What will determine how much this will change? What is the quantity that is, I mean, if this quantity is more yellow, do you expect this to go faster or slower? Faster. So what is determining that? What is plotted here? What are the colors? This quantity here is what we derived before, right? So let's say that we look at this beta in some unit of time, okay? In some delta t. This will be proportional to what? It will be proportional to S of beta prime given beta, right? Do you agree? Because the more yellow this is, okay, the faster will be evolution. Okay? But what is beta prime? So we have the delta beta is proportional to S beta prime beta. Okay. Let's say, so now this is like a difference of betas, this is a rate. So this is something times delta prime. Okay? Do you agree? So now what is beta prime? The new beta, right? But the new beta is beta plus delta beta. You agree? So what we have is that we have the delta beta over delta t is proportional to S beta plus delta beta. Okay. Now, when I have deltas, what do I want to do? Usually, take the limit. This is a no. Take the limit. It's clear. Right?
It's obvious.
In Italy, no. Okay. So you want to take the limit, right? So these things, when we take the limit, becomes what? Becomes d beta dt, which is the quantity we want. And now this quantity here, what is it? Well, you can expand this. Right? So this quantity is S of beta beta plus now the derivative of S beta prime beta over d beta prime evaluated in beta prime equal to beta. Okay? Times delta beta. Okay? Now, what is this quantity? This is the growth rate of an invader with the same rate as 0. Okay? So this is telling us that what determines evolution is this quantity here. That what we expect is that the derivative, that this quantity here, right? And this is of course a bit, but this quantity here is what determines the speed of evolution. Do you agree? So this quantity here now, if we go back to what we found before we had that S beta prime given beta was equal to beta prime eta prime plus omega minus beta prime beta plus omega. And therefore, evaluating this thing was equivalent to evaluate this thing here, to evaluate the derivative of these things. Okay. Good. Now, what is interesting in general is that this function here, okay, is some function of beta. Okay. So what we get is that we have an equation of d beta dt, which is some function of beta. Right? Now, what is this quantity here? What is this quantity? What defines these things? This is a dynamical system. Right? It's an equation which can be whatever. Right? So it describes the dynamics over evolutionary times of this straight beta. Okay? And the way we derive this is starting from this fundamental object here. Right? So the growth rate of an invaded beta prime when the resident is beta. Okay? Okay. Questions about this?
Thinking of the parameter omega, and we can define some, because I'm trying to think whether we can arrive to the conclusion that under more stress the evolution goes is accelerated. So I'm thinking whether we can define some.
So the point is that, I mean, the speed of this, I mean, here I wrote proportionality, right? Because here you have something which depends on how many individuals, for instance, depends on the mutation rate, how many individuals you have. So there is a dependency of omega you can derive. In particular, I think that the important term there is, well, I have to check. It's the population size which increase with omega. So larger omega should be faster. Yes. No, it's the virus one. No, I'm not sure. I have to check. But the point is, in some sense, the beauty of this is that you define the demographic rules and you get the results. Okay? Other questions? Okay. So in the last half an hour or less, I want to do something which generalizes this. Okay? So the idea is that what we discovered in this case is that if we have some traits, now in principle this can be true for multiple traits, right? In this case you have beta, but you could have multiple ones. And so if you have a given trait, this quantity, and you want to look at the population, at the average within a population, this thing will be proportional to, in this case, I mean, the central object will be the growth rate of an invader with traits theta prime given theta. Okay, and this quantity is where ecology goes, and then evolution will be this thing. Okay, where you have these dynamics here. Okay, where basically you are following the gradient of this invasion growth rate. Okay, these things is called adaptive dynamics. Okay, and it allows you to get a sense of where the traits are going. Now I want to describe this or sort of introduce a simple system in which we can look at coevolution with this framework. Okay, so this is what we discovered, coevolution. So let me. Okay, so let's think again about what phage. Let's go a little bit in details, more in details of what phage do and how they work. So what phage do is that this is a phage, right? A depiction. This is like a more realistic picture. And what they do is that they have DNA, and they have like a syringe where they can inject DNA. And what they do is that they attach. They have a sort of tip, a tail, it's called, which recognize a receptor on the membrane of the bacteria. Okay? So bacteria on their cells on their membrane, they have stuff that does some function, right? Because a cell is not an insulated object from the world, but wants to take things from the outside and bring inside, or things inside and bring them outside. So you have one receptor here that does some function. Particularly, this lambda phage target a receptor that cells use to import maltose, so for nutrients. Okay. And what they do is that their tail sort of have high affinity with this receptor, and these things goes around passively, but then it attached to these things. And when it attached, there is the whole biochemical magic, and these things like sort of inject the genetic material inside the cell. Okay. Now, so one important ingredient in all of this is the affinity between these two quantities. Right? So, how good is the matching? Now, the other important ingredients that I want to introduce is the fact that this receptor is something that as a function for the host, as a function for bacteria. It's not just one stuff that they have and they don't care. It's something that, for instance, is needed to import a nutrient, maltose. Okay? So, yes. Now, an important point is that this is fully general, right? So this is true for phage and bacteria, is for instance true for COVID, SARS-CoV-2, and the ACE2 receptor on cells on which SARS-CoV-2 attach. Okay? So this is a general mechanism, right? So there is some affinity between the phage, the virus and the receptor, and a function that the receptor does. Okay? So this is a nice setting to study coevolution. What does it mean, coevolution? It means that now, imagine that that these things can evolve. Okay. Now, what is best for the phage? What is best for the phage is to have a very high affinity with the receptor. Right? The highest is the affinity, the highest is the infection rate. The highest is the affinity, the more likely it is for a phage to infect a bacterial cell. Right? What is best for bacteria? It's already more complicated because on one end, it's better not to be infected. Right? So they want to be, so the bacteria want to be similar to the phages want to be similar to bacteria. So they want their tail to be similar to the receptor. Bacteria, on one end, they don't want to be similar to the phages, but on the other end, this stuff make up as a function. Right? So they are not free to choose the structure of this as they want. Right? Because if they make it a little bit more different, they might lose the ability to import nutrients. Okay? So what is going to happen if we have these things co-evolve? We don't know. But then what do we do when we don't know something? We write equations. No, what we can do is that we can try to, of course, we can simplify these systems and get an intuitions by writing sort of a simple model. Okay? This is what we're going to do now. So first of all, the structure of these things, it's very complicated, right? So we can imagine of having one variable or multiple variable capturing the structure of the phage, tail, and the receptor, but this is going to be a mess. Okay? So let's start with the simplest option. So let's say that there is one continuous trait of phages, which is Y, which is something related to the phage tail. Okay? And then we have one continuous trait, X, which is bacteria. Let me see something, sorry, I want to see just, yes. There is bacteria receptor. Okay? And then I want to write the dynamics of this system. Now let's make the dynamic a little bit simpler. Let's imagine that the latency period is very short. So very infected cells are immediately burst. So what we want to write is the equation for bacteria and the equation for phages. Okay? but which bacteria and which phages? Right? So, well, let's imagine that we have some bacteria with a given X, okay? And then we have some phage with a given Y. In principle, we could have multiple ones, but let's imagine that we have a very simple population where there is one type and one type. Okay? So what is the equation for the growth of bacteria? Huh? First of all, logistic. Okay. You have I n of x 1 minus n of x over k. Let's say we have our omega term. And then what do we have? What is the other process? Infections. So we have minus pi n of x d of y. And then here, as I said, let's assume that these things immediately burst. Good. Right? So if written like this, there is no dependency on x and y of anything. Right? So I guess you agree with me that there must be something that depends on x and y. What do you think depends on x and y? Okay, where is the affinity going in this equation? Well, V depends, I mean, V as a label, so here I'm counting a phi, yes, exactly. So phi will depend on what? On x or on y? Both. Okay. And then something else. So this captures the fact that the rate of infection depends on both the property of the phage and of the bacteria. What other parameter do you think will depend on? We say that this receptor performs a function. Yes, depends on what? On x. So what we expect is that the growth rate depends on x. Right? Good. So now, how do this quantity depends on x and y? So let's start on r of x. How do you think this growth rate will depend on x? I mean, we don't know, but let's try to get something interesting here. So what is dependency captures? This dependency captures the fact that somehow you want to perform a function, okay? And you can imagine that there is an optimal way to perform this function. And then as you become more and more different from this optimal way, your ability to import, in this case, maltose or to grow decreases. So what kind of shape for this curve would you expect? Something like this. So this will mean that basically if you are above value, you are whatever. The idea here is that this growth rate will depend on, will somehow, yes, so it could be something like this, right? Where there is some x naught, okay? And basically there is some, R naught. Okay? So if you have bacteria have exactly that sequence, they grow at the maximum growth rate. If they start to have a different sequence, they do not. Okay? Good. And then, what is the shape of xy? Well, first of all, what should I put here? X or Y? So this is the infection rate as a function of the trait of the host and the trait of the bacteria.
What do we have in 1?
Well, it will depend on somehow the difference between the two, right? It will depend on how different is the receptor from the tail, okay? So what we can say is that this depends on x minus y and peaks around 0, right? If they are the same, the infection rate is maximum. If they are different, the infection rate becomes low. Okay? With this, you can write whatever equation you want to describe this. A simple way is to say this is R naught e to the minus, let's say, x squared over 2, x squared. I can choose this x naught equal to 0 without loss of generality, and this is like some scale Cn squared, right, which determines this thing. And similarly here, I can choose some phi naught e to the minus x minus y over cv squared. Okay, cv. So this quantity here tells us how different the host and the bacteria have to be for the infection rate to be 0, right? Or how fast the infection rate declines with the difference between the two. And this quantity, this case, determines how different has to be the receptor from the optimal one for to start to stop function. Okay. Okay. So we have all this machinery here. Then what we do? We want to study evolution. Now, in principle, we can take this system and have 100 different types of bacterial viruses. Then we discovered earlier that we can get a lot of intuition based on, yes, and invasions. Right? So what we can say is, now we have these populations, right, and we can calculate fixed points. Right? We can calculate fixed points, and we calculate the fixed point we get from this equation here. We get that n star x is equal to omega over beta minus 1 p x y. Okay, and from the first equation we get that v star of y is equal to r of x n star of x, no, r of x 1 minus n star of x over k minus omega over x1. Okay? So we have these fixed points. And then what do? We add an in the Okay? So what we do is that we add one invader. And we have this population, and what the invader will do? Well, the invader will be some mutant. Well, first of all, an invader of what? Of bacteria or phages? Who can mutate? Both. So we can have an invader of bacteria and an invader of phages. And we will have the mu and mu. Okay? Now, this will be the population of the mutant phages. We trade y mu, let's say y prime, which will have here a y prime now. But then this will infect who? Will infect the resident population. So X. And this one will be n star. Okay. And then this will be y b mu y prime, y prime. And again, with this trick, you immediately see that this equation is linear. Right? Depends on the prime. So the equation for this equation here can be rewritten as beta minus 1. And then if we substitute this expression here, what we get is omega, or we get omega in front of everything, phi of x y prime over phi of xy minus y. Okay? What is this quantity? This quantity is times b. Okay? This quantity is exactly this s y prime given x1. Okay? So it's exactly the growth rate of a mutant, of a viral mutant, a mutant of a phage, when the resident population of bacterial phages has xy. Right? Sorry, this disappears. And this quantity basically is telling us, if you look at this, the growth rate is positive if you infect more. Okay? Now, what is important here is that if you infect more, who? If you infect more, the resident population. Right? Then, in the same way, it's a word equation which looks like a little bit more complicated. What you can do is you can define this, right? Where you will have R prime, this is the mutant, this is the mutant. Well, no, sorry. Here, this you can substitute with the resident. This is the mutant. And this is the mutant, this is the resident, this is the resident, this is the mutant. Okay? And in the same way, you can define, so this is for viruses, you can get the growth for bacteria, x prime given x1. Okay? Then you just substitute these equations, you make a lot of mistakes, you do all the calculations 10 times, and at the end, you arrive to some equations which describes what? Which will be some dy, well, some dy dt, which will be proportional to ds dy prime xy dy prime y prime equal to y. Okay, and the same for pages. So you get to some equations dx dt, dy dt equal to some f xy, fv xy. Okay? Which tell you how the traits of bacteria and phages change. Right? And what is important here is that this is a dynamical system, right? So you have the change of the traits of the bacteria as function of what are the traits of both bacteria and phage. Is this clear? Not the calculation because we didn't do it. But is it clear the setting? Okay. So you do this equation here, and what you discover is that, well, suppose that you have a dynamical system here. What do you do if you have a dynamical system? The first thing you do, calculate, because you already saw this. You calculate fixed points, right? And then you check there. Exactly. Right. So you can calculate fixed points, and if you calculate the fixed points of this, what are the fixed points of this? What you discover is that the fixed points of this, which you can calculate setting these things to 0, what do you think is going to be the fixed point here? x
So equal what?
x equal to x naught, and y equal to x. So the fixed point here is x equal to y equal to x naught. What does it mean? That fixed point corresponds to the case where the bacteria are at their optimal receptor and phage match them. Okay? Right. But then, is this the end of the story? Why? Invasion, this is the way this is calculated. But if you have a dynamical system, you calculate fixed points and then you stop, or you do something else. What do you want to know about fixed points? Whether they are stable. Right? So then you check stability, and what you discover is that this is stable if you have these two parameters, right? And this is stable if Cn over Cv is lower than some pressure. Okay? So basically, if these things, so if they, what does it mean that this is lower than some threshold? It means that these things is very narrow. Okay? Which means that for the bacteria, they are very constrained in how they grow. Right? So if they're very constrained with respect to the effect of infection, then what happens over evolutionary times is that bacteria have to stay there. Okay? But then this is unstable in this case. Okay? So this means that over evolutionary times, if this condition is not true, okay, so if we are slightly more permissive, then what we expect is that it doesn't go to the fixed point. And what you discover is that if you look at the dynamics on the xy plane, you have this fixed point. And what happens is that there is a limit sample. So you keep cycling. Okay? Which is what? Which is basically saying that bacteria evolve their receptor to escape from phages. Right? And phages are following. Right? Now, this distance here is determined by this ratio, okay? Because bacteria are constrained, right? So they want to be at the sweet spot where you are enough away, you want to be away from this in order to be able to escape, right? But not too far, because otherwise you don't function. Right? And so this creates this arms race where basically there is continuous evolution, right? There is continuous evolution for the bacteria to escape phages and for the phage to follow bacteria. Right? And this is called sometimes red queen effect, okay? From the Alice in the Wonderland. Mm-hmm. That basically you keep running without going nowhere, right? Because the idea here is that as they co-evolve, they will be like at some fixed difference between them. And so they keep changing over time, but actually nothing is changing in the system. Right? Because they are continuously changing, but actually the bacteriophage keep infecting at the same rate. Okay? And so, yeah.
The fact that the virus, the phage is evolving at much faster rate,
it impacts what is this threshold. So the way this rate enters is in this.
You cannot be in an extreme case in which you disrupt this.
Yes, I mean, if, let's say, the difference in the evolution, the mutation rate affect the speed. So basically it becomes less, the sort of width of this has to be much larger proportionally in order for this to become convenient. Okay. Because if you stay here, the difference is going to be 0. If you stay in this circle, the difference is non-zero. Right? And the difference you get, and this depends on how far you are. Questions? Now, this is in a very simple setting where this is sort of this receptor dynamics is in one dimension, in reality, this is in a very high-dimensional space. But actually, the phenomenology that you see is similar. So, for instance, if you look at flu, the virus of influenza, of grippe, that thing is continuously mutating and our immune system is continuously mutating to discover it. And so there is this continuous dynamics of the virus, in this case, mutating away of our immune system. This is slightly different. So the virus is mutating away our immune system, and our immune system is tracking this virus. Okay? And yes. Important point. So what is the important message here? That when you think about evolution, don't think, one example of something that is steadily optimizing something else. Right? So there is no obvious, in this case, there is no obvious function that this system is optimizing. What evolution does is creating dynamics. Right? In general, a dynamical system is not a system optimizing something. Okay? So it's a force that is local in another way. So evolution acts on the traits given the current environment. Okay? But the current environment is set by the traits themselves. So where evolution drives the system to depends on what is the current condition. The current condition of the environment. Okay? Plus the constraints. Okay? Over. So thank you very much. I really enjoyed staying here with you. I'm very, very thankful to the organizer for having organized this. And we'll cross our path very soon as we go to the